Hello, everybody. I'm Spencer. I'm a graduate student here on campus, and I'm part of an organization called Project Bridge. And I'm here today because I'm hosting the Postdocs Week's a Gong show that they're doing today, and really excited to be here. So for you guys, we're just going to hit a couple of ground rules on what a Gong show is, and then we have some great speakers who are coming up. And this is also after they've gone a place where if you want to give your own luck at like trying the speech that I'm going to explain, you can. There are cash prizes or valuable prizes on the line. Bruce says yes. So it might be worth coming up here and trying your hand at a 3-minute talk. So yeah, the Project Bridge has been formatting this Gong show for a while. That's why you all are holding cowbells. Basically, what you're going to be doing is doing a 3-minute talk about your research or a scientific subject that interests you. And you're going to try to explain the whole concept in 3 minutes and not use any jargon. And if you do hear somebody use jargon, even if it's jargon you think other people might know, as an audience member, your job is to ring cowbells at them, and they have to stop, go back and explain the jargon that they actually thought they didn't need to explain, and then try to continue on. So be liberal with these cowbells. Part of the fun is that you get to essentially go and like publicly shame your fellow speakers. So get involved, and it is always more fun when we do that. So yeah, without just a reminder on the prizes though, there is a voting system with a bunch of QR codes that are somewhere. There's also an email link. So after you've heard everybody talk, definitely vote so that you can like give whoever you think gave a really great talk a gift card and you just a little bit of like bragging rights among a very select group of people. So yeah, thank you guys so much for all showing up. I am going to probably get us started then. And then just for speakers, if you look for me over in this corner, I'll give you a 1 when you're 1 minute away. And then this is 30 seconds, but you're not heavily timed. Nobody will cut your mic off. And then after you give your talk, we'll leave a couple of minutes if anybody wants to ask questions then, and then we'll move on to the next talk. So it's not just talk after talk. So without further ado, then I'm going to introduce Allison Swain, and her talk is going to be titled "The Secret Life of Yeast That Lives on You."
Hello, everyone. Thanks for coming today. So raise your hand if you've ever had a yeast infection. I'm just kidding. You don't have to admit to that. The reality is 3 in 4 women get at least one yeast infection in their lifetime. So a lot of us are very familiar with that and how much they are awful. So the yeast that infects us is actually the yeast that naturally lives on us throughout our lives. And they don't really harm us. They just kind of live there and hang out. And for healthy people, it's not a major issue. The issue comes in immunocompromised people. So that's people with like a non-functioning immune system. They have AIDS or they're on chemotherapy or immunosuppressive drugs. And these people can actually get bloodstream infections with these yeasts. And if you get a bloodstream infection, you have about 50% chance of dying from it. So it's really a serious issue. So these yeast, are living on us and they can infect these people that have a bad immune system. The yeast typically looks kind of like this. They're just like a round yeast cell, they bud, and this yeast can divide asexually. So they just make copies of themselves. Thank you. They make copies of themselves with the same DNA and replicate in that manner. So this yeast can only kind of mate in that asexual way, but they do a cool thing called a phenotypic switch. So that means they can kind of switch how they look without changing their DNA. And they do that by turning on and off different genes that they express in the cell. So the switch that I study is called the white opaque switch. And they can switch between these two states. This state does not mate, but this state can actually mate sexually. And so when they switch to this state, they can undergo sexual mating. So I found a really cool enzyme in my work that can change the genes that get expressed in each of these cells. And it does that through modifying the messenger RNAs that get produced from these genes. And so I'm studying how these messages get modified, which messages get modified, and how they regulate the switch from going back and forth between these two states. So thank you, and come talk to me about yeast if you are interested. Yeah, if anyone has any questions, I'm happy to answer anything. Great question. So yeah, Bruce asked, what's the purpose of this research? So, the more we kind of learn about the biology of these yeast cells, we can kind of figure out different ways that we can treat them. And so this white opaque switch not only regulates the mating pathway, and mating is interesting because when they can do sexual mating, they can evolve faster. So acquire new traits, adapt to the drug treatments better. So besides the mating, these white and opaque cells also have different virulence traits, and they can live in different parts of the body. So if we could figure out a way to regulate this white opaque switch, we might also be able to kind of treat these infections a little bit better. All right, thank you.
Thank you again, Allison, for a great way to open this up. Also, just to note, the reason we keep doing this is we've been told not to jump straight up this ledge. If you do end up coming up and giving a talk, walk around on either side. I think it's a safety or security concern, so we're putting in our steps. So for the next talk, we have Deb Nandy, and his title is going to be, "But wait, is that real or fake news?"
That fine morning, as I was sipping through my warm cup of herbal tea, my phone buzzed with some notifications of this wonderful app that I have installed recently called Real Fake Science. I looked and I was mesmerized by 5 scientific discoveries that are sensational. Number 1, butter. One should consume as much butter as one likes because that helps keeps the inner walls of the veins and arteries lubricated so that the blood can flow smoothly. Number 2, paleo diet. Paleo eaters actually consume lesser number of calories throughout their lifespan because just like our ancient cavemen, they also pass away in their early teens. Number 3, gasoline. Why do gasoline smell so good? Because gasoline is a fuel. So that's why when you smell it, it burns through all your brain cells that actually make an individual think. Number 4, excuse me, my doctor friends here, stethoscope. Why do doctors put stethoscopes on different parts of a human body? Because sometimes they forget where the heart is. Number 5, dinosaurs. That's my favorite. Why do dinosaurs die, or how did they extinct? Well, they exhausted all the food on the earth. They started digging holes for food, and unfortunately, they all got stuck in those holes. That's why we find dinosaur skeletons underground. Well, exactly. This is the problem. This is 21st century, and we have at one hand the blessings of scientific inventions and technological advancements where we can get huge amount of data, but at the same time, statistical analysis of those data to come out with meaningful inferences has been put in extremely highly challenging situation. As a statistician, or the lovingly called data scientist in these days, I dig through those data, I discard the fake news, and I bring what is the absolute truth to the human beings and common public. Imagine my wonderful team of scientific collaborators are working on a project studying a rare human disease, and they have collected lots of data on biological factors that they think might influence which individual would get the disease or not. As a statistician, I perform some humble statistical tests, and I compare for each factor whether the value of the test passes a threshold or not, right? And then if it does, we declare that factor to be statistically significantly associated with the rare disease. As simple as that. But imagine if that threshold is too stringent, then many of the potential discoveries will be missed. We call it as false negatives. But if the threshold is too lenient, lots of things will pass the test, which might not be actually true. Remember those butter gasoline dinosaurs? Those are called false positives. As a researcher, my current research involves developing those appropriate thresholds so we can minimize the number of false positives and false negatives. In other words, I scavenge through big data, find out the real signal. Okay. By the way, I will end with one news that came up in the Real Fake Science this morning. Scientists have confirmed, statistically speaking, human beings who have ever attended a Gong show in person are more likely to choose a butterscotch flavored ice cream compared to those who have never attended one. But wait, is this real or fake news? You will tell me that. Thank you. And right. So that's why there are different branch of statistics where we choose different measures to empirically derive, driven by the data, that what should be that threshold, right? So that these two numbers, the false positives or false negatives, are minimized. For example, we can do, and again, like, I hope that I'm free to use some jargon here, but then there are different ways or branches of schools of statistics, like Bayesian approach, right? And then there is another approach like classical approach. So people have been attacking this problem in different approaches to see which one in the simulation studies, for example, where we know the actual truth, gives the best result. So that way we can say in the common public that this approach, if you use it in this context, this will give the best results. And one approach might not be the one size fits all approach for every data sets. Yes. Right. So thank you for pointing that out. So that is what we call correlation is not causation, right? So that's why we have a whole bunch of statistics where we take advices from the experts called causal inference. So that causal inference exactly does that by setting up the experiments like as a randomized clinical trial, like we do in clinical trials, where we can randomly assign people in the healthy group and the treatment group like that, and then apply techniques from the causal inference branch so we can actually target exactly what you said, which is correlation is not causation. Thank you. Thank you.
Oh God, it's hot in the sun. Okay, that was a great talk. Thank you so much, Deb. We've got another prepared talk here coming from Satya, and his talk is going to be tracking you down with needles. So thank you, everybody. Welcome him up here.
It'd be better if you left it on there. Thanks.
Oh, sorry. Miss Gary, I don't have so good news for you. Your breast cancer has recurred, and we are having set of treatment plans, but then we have to actually know what's going on in your breast cancer. So we have to perform biopsies or even radiology to assess the state of the cancer. Dr. Cooper, I'm not so comfortable with having biopsies. It just gives me uncomfort. And even the radiology. Do you have any other measures to actually assess the cancer? Cut down to the successful completion of my research, and then the next visit of Ms. Gary with Dr. Cooper. Ms. Gary, I do have good news for you in terms of the radiation or biopsy related kind of assessment. I ran into research of a postdoc, Satyanarayan Rao, and it was really cool in my opinion. And let me just explain how cool it is. So imagine we all are made up of cells, and they have very fundamental unit called DNA that is same in all the cells, but they are different in the way they are actually organized. So to help you explain, let's just imagine that you have this string that is laid down here, and you have two different kind of proteins. One is bigger in size and one is smaller in size. And they are actually dancing based on the hormones that you secrete. So for example, in your body, you have estrogen hormone. We don't have that. But based on that, basically, this particular small protein has gone rogue and making cancer cells in your body. So what Satya has done really cool is that it is a fact that cancer cells grow very fast. And then, so by that result, actually, they also die very fast. And they release this string of this DNA bound by these small and large size of proteins in the blood. And what Satya is doing is basically tracking them down just from the blood test. So let me explain what is happening. It's like, let's say in your sister who does not have breast cancer, here, that can actually tell that in your body it's binding here, and in your sister's body it's binding there. That's how they can track the cancer. And I find it very cool because every next visit I can actually find where all this rogue protein is binding in your body, and that way I can actually assess your cancer. And then I can actually design better treatment plans for you with the drugs that we have available on the stage. Thank you. Any questions? It's actually better because blood is a pull down of all the microenvironment of the cancer. So in the biopsy, you have a slice that's very local. But in the like blood draw, you have the snapshot from all the microenvironment also. So it's better.
Thank you.
Thank you again for that great talk. So as I mentioned at the beginning, after these talks, we opened it to the floor to anybody who would be brave enough to go and give this a shot themselves. And we've already had one volunteer. So this is really exciting. We're going to be welcoming up Stephanie with her talk titled The Pooh and You.
You guys are really far away. Okay. All right. So has anybody pooped ever? Oh, some of you need to see a doctor, like ASAP. Okay. Well, I hate to break it to you, but all of you are infested. You and you and you and everybody there and me. You're all infested with trillions of bacteria. They're on your skin, in your mouth, in your airways, and in your intestines. And these bacteria are vital to the way that our bodies function. This is called the microbiome, which are all the bacteria that live anywhere in your body. And in particular, the bacteria in our gut play an important role in how we digest food. So that means that two people eating the same diet, having the same food intake, they can respond very differently to those nutritional intake just because of the bacteria that are in their guts. There's so much information that we can learn from poop, and yet we just throw it away. This is a diaper from my son. He'll be 2 next week. It's very hot out, so I don't recommend opening this bag because it's been fermenting since this morning. But there's so much information we can learn about him from this dirty diaper. So if anyone wants it, I'll just leave it here for you. The composition of our gut bacteria is influenced by a lot of factors, including what we eat, who we live with, our environment, and our overall health. As babies, the biggest impacts on the gut microbiome are how we are born, so either vaginally or by C-section, what we eat, so either breastfeeding or formula feeding, and whether or not we receive any antibiotics, so if you had an ear infection or a pneumonia. But there are smaller factors that can influence our gut microbiome too. Because the microbiome in adults and older children has been shown to be affected by obesity, and because the maternal microbiome is known to help colonize or populate the infant's microbiome, we asked the question whether maternal obesity or overweight status during pregnancy could influence the infant's gut microbiome early in life. Infant poop samples were collected at 1 month of age. I was luckily not the person that had to go through those. And the different bacteria were collected. And we found that relative to infants with moms of normal weight, the infants from mothers with a higher weight had lower counts of a bacteria that produce a chemical called short-chain fatty acids. Short-chain fatty acids are small molecules that are produced by bacteria in the colon from dietary fiber, and they're part of the reason that tiny bacteria can affect the entire body, including appetite, glucose regulation or sugar metabolism, your blood pressure, and also your appetite. There's growing evidence that short-chain fatty acids impact overall health and may protect against the development of obesity. So giving short-chain fatty acids to mice actually protected against weight gain when they were given a high-fat diet. And in humans, giving short-chain fatty acids to adults with type 2 diabetes lowered their blood pressure into a normal range. So while we're not sure what it means for these babies to have lower prevalence of short-chain fatty acid producing bacteria, we did show
that maternal weight status during pregnancy influences the infant gut microbiome. So thank you for coming to my crappy talk about poop. I hope you enjoyed. Oh, you want this? Is that one? Yeah, so the question was, can we change the microbiome? And is that something that we could do in terms of intervention? And the answer is yes, the gut microbiome is modifiable. So for example, let's say if you were on a plant-based diet and then decided, oh, hey, I love McDonald's, they're having this great sale, I'm gonna only eat McDonald's for the next 24 hours. Even after just those 24 hours, your gut microbiome is different. So it really is pretty easily changeable. The hard part is to know exactly what to put in there, and is that different for different people? So a gut bacteria that might be helpful for me may not be the same thing that's helpful for you. There's a new probiotic out that's specifically geared toward infants to provide them with a specific bacteria called Bifidobacterium infantis. And hopefully that they've shown that it might reduce colic, it might reduce fussiness and things like that, which any parent will tell you if you could get your baby to cry less, it's probably worth it, no matter how expensive it is. So the question is, should you actually take probiotics in order to impact your diet, and are they worth it? Yeah, or just eat a lot of yogurt. I mean, I'm a big Noosa fan, so I eat that a lot. But I think probably overall, your best bet is just to eat lots of fruits and vegetables and stay away from a processed diet rather than try to supplement with probiotics and bacteria that may just actually get digested by your stomach acids. But there is some evidence that taking probiotics can ultimately impact your gut microbiome. So probably in the future, I could see us having therapeutics and maybe there'll be like, some sort of like Activia lean yogurt that you could eat. But for now, I would just probably say eat your fruits and veggies.
Thank you, Stephanie, again for that great talk. I'm going to put out one more chance for somebody to come up like Stephanie did and give an off-the-cuff talk. Reminder, there are both real rewards for giving talks that people vote for, and it's a lot of fun. We've enjoyed it so far as audience members. It is, I know, scary and intimidating. Man, now I just put them on the spot and nobody liked that. I shouldn't have done that. But yeah, these have been some great talks. If we are reaching near the end of the talks, there are QR codes that I think are going to be passed out. There's also a link in the email address. The quiz or the voting thing that you have started only with the people who gave us both talks and titles at the beginning, but there is a section labeled Other, where you can put either the speaker's name or the title of their talk and vote for the other people who have also been given some great talks up here. Definitely vote. This will get them real money in the form of gift cards. So use your nepotism, vote for yourself. And then additionally, there is also going to be lunch that's going to be over here in this little tent over here on the far left. And so you guys should all come and eat some lunch. The last thing I'll kind of mention here is if this type of communication was really engaging to you or you really liked it, there's an organization on campus called Project Bridge that does these very regularly. It's actually started by a postdoc several years ago here named Erin Golden, who was a postdoc in the Barlow lab and has since moved on to other things. She likes to say bigger and better, but she left us and we're sad, so we just say other things. But yeah, so postdocs and grad students, it is designed to have early career scientists meet with the community, with government, and really kind of try to bridge that gap between the ivory tower and the spaces around us. So if you're interested in that, feel free to come and find me during the lunch or look up Project Bridge Colorado, and you'll find our website. Thank you guys all for participating, both as audience members and as speakers. This has been really great.